discovered the cell, as the chapter is fundamental unit of life. So Robert Hooke was the one who has discovered the cell in 1665. He has discovered a cell from a cork which is used mainly in the champagne bottle. You must have observed the bottles of the wine as well as the champagne. They are covered with a tight pressure with that help of a cork. Now, cork is really very hard, so it is also very difficult to take a small section of the cork, but he was able to do that. How he was able to do that? He boiled that cork and cut a very slight section of it. By boiling it, the wood becomes little softer. During the time of 1665, there was no such technology of the microscope. So he designed his own microscope, which looks like this. If you can see clearly, the microscope, it is very small and gentle, just like a bottle. The outer part, the top part is of little bigger size, and the point at which we are focusing it is small. That means we are focusing on it. So when he cut the section of a cork and observed it, what he observed that it looks like a honeycomb. You can see the image. From the left side, the cork, it comes from the bark of a tree. Robert Hooke took that part, observed it, and the structure which he observed, it was just like the right picture which is shown on the slide of a honeycomb. So he gave them the names of cells. Now, what are unicellular organisms? What are multicellular organisms? Because ultimately the cell, it is a building block of life, and it will comprise of so many things. We are going to cover that. As in class 8, what we have studied, that a group of cells will form a tissue, a group of tissue will form an organ, and a group of organs, they will form an organ system and ultimately an organism. But there are some organisms which are single cell organisms. We call it as a unicellular. Unicellular means only one cell organism. Example, basic example, amoeba and chlamydomonas. Multicellular organisms, we all are multicellular organisms, humans, animals, fungi, plants. And different cells which are there in our body, nerve cells, fat cells, bone cells, sperm, ovum. Now, cell. What is cell? It is the smallest unit that is capable of performing life functions. All the functions which are going in our body, which are taking place in our body, the cell is responsible for it because it is the first step. Now, what is a basic cell made up of? As I have told you that the simplest unicellular organism is amoeba. What is amoeba form of? If you see, I'll show you at the back slide. The left picture, amoeba. Can you see it's a simple organism? It has one membrane, nucleus and cytoplasm. That means for any cell to survive, it should have plasma membrane, second, nucleus, third, cytoplasm. Now, first we are going to study about these three because they are the basic organelles, basic part of a cell due to which a cell is living. After that, we are going to cover about the organelles. Now, what is cell? The shape and size of the cells are related to specific function they perform. The cell, the shape is confined for a particular function. For example, if I say red blood cells, they are concave in shape, biconcave in shape because they have to be circulated into our body. If they would be pointed ones, then it would be very difficult to get circulated. So shape and size is dependent on the functions they perform. Second, each living cell has a capacity to perform basic functions. Cell contains a specific component called as organelles. We are going to study about various organelles. Each kind of cell organelle performs a specific function, and all cells are found to have a same organelle irrespective of their function and which organism they are found. What it says in short that each and every cell is prescribed with the particular function. It does not mean that RBC will do the function of WBC or WBC will do the function of the platelets. So each and every cell, they know which function they have to do. And for that, they have various organelles which help them to do that. Let me give you a very basic example. Suppose this is your complete classroom. This is a complete classroom, right? We can say that this is a cell. Your classroom is a cell. You all are the various organelles. Okay? Supposingly anyone is mitochondria, anyone is ribozymes, anyone is Golgi apparatus, that we all have studied in class 8. And there is one nucleus. Who is your nucleus? Your teacher is a nucleus. And all the students, they are the organelles. So, your teacher, which is a nucleus, is responsible for giving you particular function and task. And you people are responsible for doing that. And when all of you with me, that means all the cell organelles with the nucleus, when perform that function, then that cell will perform perfectly. Am I clear? Now the first basic point of the cell is cell membrane or plasma membrane. It is the outermost covering of the cell that separates the cell content from its surrounding. It is very important. If there would be no outer covering, all the material which is inside the cell, they will be thrown out of it. And that we don't know. And we don't want that. So what is the basic function of the plasma membrane? It allows the entry and exit of material in and out of the cell. Now you will ask, ma'am, all the particles can go in and out? No. There are some particular particles which will go inside and out of the cell because it has a semi-permeable membrane. Semi-permeable means which will allow entry of some and exit of some. Right? Now plasma membrane is really flexible. It is not very hard. That's why it is made up of proteins and lipids. Now, the movement of particles mainly which are gaseous phase, it goes in and out of the plasma membrane to the cell by the process of diffusion, which is a gaseous exchange. It takes place within the region from high concentration to low concentration. That means they have to maintain an equilibrium. The movement of the particles, mainly gaseous particle, will be from high to low until and unless they both will create an equilibrium. Now there is one difference between diffusion and osmosis. Osmosis is exactly same, that is the movement of particles from higher concentration to lower concentration, but the only difference is water molecules. In diffusion there is gaseous molecules, whereas in osmosis there is water molecules. The movement of oxygen and CO2, the exchange of oxygen and CO2 inside the alveoli of our lungs is taking place through diffusion. When the concentration of oxygen is high, then it will relax, right? And it will contract so that the CO2 will go. In the same way, similar vice versa. When CO2 concentration is high, it will move to the lower concentration of CO2. Similarly, when O2 concentration is high, it will move to the region which has the less O2 concentration. Same way in the osmosis, the movement of water within the cell, it is from higher to low. You can see in this figure, there is one beaker which is given, and in this beaker there are few particles. On the left side, there is high water concentration, and there is, if you see, no sugar concentration, or we can say very low sugar concentration. On the right side of the beaker, you can see that there is high sugar concentration and low water concentration. And there is one selectively permeable membrane. So the movement of water will be towards left to right because there is low water concentration over there. But because why? The movement would be from higher concentration to lower concentration. So what is the basic difference between diffusion and osmosis? Diffusion, it can occur in any medium, whereas osmosis, it can only occur in liquid medium. Secondly, It does not, diffusion, it does not require a semi-permeable membrane always, but osmosis, it will always require a semi-permeable membrane. And third, we have discussed that diffusion is a movement of gases, whereas in osmosis, there is a movement of water. Now, when we are talking about osmosis, there are three types of solutions which we are going to study in osmosis depending on their concentrations. The first one is your hypotonic solution. Now, since I told you there are three types of solution and it is little very difficult to remember that which solution is responsible of which activity, there is a small method which I have learned in my school and I want to share it with you as well. Hypo means hippo. Hippo means anything which is very fatty. That means ultimately the resultant cell will swell up. It becomes fatty. So let's begin. Hypertonic solution is a solution surrounding the cell which has higher water concentration in the cell. Right? Now water molecules are free to move across the cell membrane in direction. But more water will come into the cell than will the outside atmosphere. The net resultant that it would be that water will enter into the cell and cell will swell up. In short, we'll say the concentration of water is less and the concentration of water inside the beaker is really high. So, the movement of water will be from higher to lower, that means from outside to inside, and the cell will swell. So, how to remember it? Hypo means hippo, that means cell is going to swell. If cell is going to swell as a result, so initially cell would be having less concentration and water outside the cell would be having high concentration. So ultimately the cell has to be swelled up, so water will go from outside to inside. Fine. Second is iso. Isotonic means equal. Equal, that means the concentration of water inside the cell as well as the surrounding medium, they both are the same. So there will be no net movement. The water goes in both the directions and an equilibrium, maybe not, but there will not be any high movement of air or water. Hypertonic, so it is just opposite to the hypotonic solution. That means the concentration of the cell, the water concentration inside the cell is initially high and the surrounding concentration of the water is less. So, the movement of water from high concentration to low concentration, that is from inside to outside. Next topic is plasmolysis. Now, what is plasmolysis? It sounds similar to the osmosis, right? Now, plasmolysis is exactly similar. The difference is that it is a phenomena by which the plant cell shrinks away from the cell wall when it loses water by osmosis. Now, this time you have to remember your class 8. In class 8, we have studied about plant cell, that there is two membrane in the plant cell. One is the cell wall, which is the outermost membrane, and inside there is a cell membrane, that is plasma membrane, which we have already studied. Now, I have told you that plasma membrane is a flexible membrane, right? Why? It is made up of proteins and lipids, whereas your cell wall, it is really rigid, very high and thick membrane because it is made up of cellulose. That's why it is present in the plants, because we people, animals, do not have cellulose in our body. So, when the plant cell, in plant cell, there is a movement of water inside and outside. The cell wall will stay intact. But initially, when the plasma membrane, it can shrink or expand, depending on the concentration of the water. So, cell wall, it will stay intact, and inside the cell membrane, it will shrink or expand. So that shrinkage of the cell membrane is called plasmolysis. You can see in this picture on the first, on the first picture there is water molecules, and in the plasma membrane and cell membrane, right? Two layers are there. The outside layer is cell wall, and the layer which is next, that is plasma membrane. Ultimately, when there is a shrinkage, the vacuole will shrink and that's why the membrane will also shrink. Cell wall. Just as I have told you in the last slide, that it is a hard outer covering of the cell which is present outside the cell membrane. You all can see it is composed of cellulose. Now it is a complex structure and it provides structural strength to the plants. If you see the plant, they are a little bit harder, right, in comparison to animals. We are soft because we don't have the cell wall. As I have told you, we don't even have the cellulose. Cell wall is present in plant cell and help the plant to withstand the environmental changes. Definitely, we people always stay in the environment which is suitable for us. We people have our home. We are not making that harsh condition of the environment. But when it comes to the plants, they are facing hard conditions like rain, sunlight, wind, cyclones, too many things. So the cell wall, it will help them to bear those harsh conditions. Now nucleus, I have explained to you as well. Nucleus is the main coordinating center of a cell. We can also say that it is the brain of a cell. It has a double layer covering called nuclear membrane. Nuclear membrane has pores which allow the transfer of material from inside the nucleus to the outside. Nucleus is most of the time is present at the center. Why? Because it is responsible for sending the signals to all the organelles so that they will get the information and they can do your work, just like our classroom. I am the nucleus, your teacher is the nucleus and you all are the organelles, and I am responsible for telling you the work. In a similar way, the nucleus is responsible for telling work to various organelles. Now, your nucleus, it contains chromosomes which are visible as rod-shaped structure only when the cell is about to divide. Chromosomes, it contains the information for inheritance of features from parent to the next generation in a form of DNA. Now, DNA is deoxyribonucleic acid, deoxyribonucleic acid. It is responsible for containing all the information of your body. That's why it contains main genetic material. Chromosome contains the DNA. And when there is a division of the cell, the chromosome, they divide. Initially, in the nucleus, there is a genetic material in a form of a chromatin, which is a thread-like structure. It will convert it into chromosome only when the cell is ready to divide. Otherwise, it will stay in a thread form. Cytoplasm. I have told you the cell is mainly comprised of three things. The first thing we have studied, that is plasma membrane. The second, and in plasma membrane, what we have studied about diffusion and osmosis. The second thing we have studied, that is nucleus, and the third most important, that is cytoplasm. Now, your cytoplasm is a fluid content inside the plasma membrane. It contains specialized organelles. Main function, it helps their organs to stay at their particular positions. Now, those three were the most important. Now, we are going to cover about the cell organelles. That means we are going to cover about what all organelles are there in the cell and what are their functions. And you will get to know that all the important functions will be done by students. Why students? Because you all are cell organelles. Okay. So, in cell organelles, we are going to study about endoplasmic reticulum, Golgi apparatus, lysosomes, mitochondria, plastids, and matrix. So, let's first begin with endoplasmic reticulum. In short, we call it as ER. ER is a large network of membrane-bound tubes and sheaths. It extends throughout the cytoplasm. There are mainly two types of ER, rough endoplasmic and smooth endoplasmic. Rough endoplasmic reticulum is responsible for the synthesis of, tell me? Rough endoplasmic reticulum is responsible for the synthesis of proteins, whereas smooth endoplasmic reticulum is responsible for the synthesis of lipids. The name itself suggests you. If we talk about lipids, what are they? They are some oily consistence. So they'll be smooth. And proteins are here. They are protein. They are not so smooth. So remember, rough means protein, smooth means lipid. Another function of the ER, it serves as a channel for the transport of material between various regions of the cytoplasm and between the cytoplasm and the nucleus. Why? Because ER is present just below the nucleus. Therefore, it is the first closest organelle to the nucleus, where the nucleus can sense the signal. It functions as a cytoplasmic framework, providing a surface for various biochemical activities. Another very important function of the ER is also do the detoxification. Next is Golgi apparatus. Once the synthesis of the proteins and lipids have been done by the ER, those material will go to Golgi. It was first discovered by Camillo Golgi. What is Golgi? It consists of a system of membrane-bound vesicles arranged parallel to each other in stacks called cisterns. The material synthesized near the ER is packed and dispatched to various targets inside and outside the cell to Golgi apparatus. So, just I have told you, the protein and the lipid which has been synthesized by the ER, it will be catched by the Golgi apparatus in a form of vesicle, just like bowls. Okay, and it is responsible for storage, modification as well as packaging. It is also involved in the formation of lysosomes. We'll study that further. Lysosomes. Lysosomes is called as the waste disposal system of the cell. Why we call it lysosome is a waste disposal system of the cell? Because it helps the cell to clean it by digesting any foreign material as well as worn out cell organelles. Definitely, if the cell is not going to die or an organelle is not going to die, the person or an organism will keep on expanding, right? But we stay in particular size and shape. So there is also a system in cell as well. Now, how it do that? It contains a powerful digestive enzyme which is capable of breaking down all organic material. Okay. Now, when the cell, point is that it is also in a shape of a bowl. Okay. It contains that enzyme. So any material as well as the Golgi is responsible for sending the waste organelles to the lysosome, as it contains the digestive enzyme. So anything which comes to lysosome, it will be eaten up by it. But it is also called as suicide bags. Why we call lysosomes as suicide bags? Because if anything happens to the cell, the lysosome will break and the digestive enzymes which are present in the lysosome will eat up the cell content, organelles. That's why it is called as suicide bags. So that's what written. When the cell gets damaged, lysosomes may break open and the enzymes digest their own cell, thus known as suicide bag of a cell. Mitochondria. Mitochondria we all have studied in class 8, the very basic line that it is the powerhouse of the cell. Why we call it the powerhouse of a cell? Because it is responsible for generating ATP, which is the energy currency. ATP, adenosine triphosphate. It is responsible for providing energy to the cell. Now mitochondria is very intelligent. Why? Because sometimes it does not require the information by the nucleus. Just like we can take our class. What is our classroom? It is a cell. Who's the nucleus? Your teacher. You all are organelles. But there is one monitor of the class as well. That monitor we call the mitochondria. Initially I am responsible, or nucleus is responsible for everything. But sometimes it happens, anything happens in a class and monitor takes the decision. In a similar way, the mitochondria, when mitochondria senses that cell is less of energy, it secretes ATP without taking the permission from the nucleus. Then that's why we'll say that it has its own genetic material in it. It has some bit of information due to which it is able to send the signal and do its work. Mitochondria has two membrane covering. You can see it on the figure as well. Outer membrane is very porous, whereas the inner membrane is deeply folded. You all can see in the image how it is folded, and that's why it is called as cisternae. Mitochondria are able to make their own protein as well as they have their own DNA and ribosome. That is why they are able to produce ATP when it is required. Second, plastids. Now, plastids, they are present in plant cells. Two types of plastids are there in a plant cell. Chromoplast and leucoplast. Chromo means color. Leuco, opposite of color, colorless. Now, plastid contains green pigment chlorophyll. That's why they are called as chloroplast. Leucoplast, they store the material such as starch, oil, and protein granules. Your chloroplasts, they are mainly present in leaves and fruits. Because they are chromoplasts, they come in three different colors: green, red, yellow. Three basic colors, and the combination of red and yellow colors gives you different types of a color in the plants as well as the fruit. Leucoplasts, they are mainly present on the roots of the plants. That's why they store starch, oil, and protein, all things which is required for the food requirement. Plastid contains the membrane layers embedded in a material called stoma. It has a similar structure like a mitochondria because it has its own DNA and ribosomes. That means the plastid has also the power of taking its own decision. Vacuoles, the main last organ, that is vacuole. It is called as a storage sac for solid and liquid content. If you see that in plant cell, if you remember your class 8 in plant and animal cell, you see the biggest portion of a plant cell has been taken up by the vacuole. Why? Because it contains the material. And in animals, there are very small vacuoles, very small size of vacuoles, but many vacuoles. In plants, there is one vacuole, one or hardly two, but that is of a very big and large size. In plant cells, they are full of cell sacs and which provide turgidity and rigidity to the cell. Rigidity, stiffness, turgidity, water balance. Vacuoles, they store amino acids, sugar, and various organic acids and some proteins. That means it is a storehouse of the food and energy. In amoeba, the food vacuole contains the food item that the amoeba has already consumed. That means it also contains some waste material in some organisms. Vacuoles play an important role in expelling excess water and some waste from the cell. So it is also doing some small function of cleaning up the cell. Now when we come to the basic, then the last, once we have studied all the organelles, so let's come to the very basic thing, that is the difference between plant cell and animal cell. Animal cells, they are generally small in size in comparison to the plant cell. In animal cells, cell wall is absent, whereas in plant cells, cell wall is present. Third, except the protozoan Eubulana, no animal cell possesses plastid, whereas in plants, all plants, they contain plastids. In animal cells, vacuoles are smaller in size, whereas in plant cells, vacuoles are larger in size. So let's do the complete small summary of this complete fundamental unit of life. That is, we have started that how the cell is called as a fundamental unit of life. Robert Hooke was the one who discovered the cell. Cell is a building block of life. He have observed it under the microscope, first via Coxon. Then we have studied how the cell is made up of. Cell is made up of mainly three things, that is plasma membrane, nucleus, and cytoplasm. Their functions we have studied. After that, we have studied what are the main organelles of the cell. 6 main organelles we have studied: endoplasmic reticulum, Golgi apparatus, lysosomes, plastids, vacuoles, and the chromoplast, which is a part of vacuole. And last, we summarize in the form of plants and some animal cells so that we can understand the function of each and every organelle and how is it responsible for making a complete life. Thank you.